Okay, so good afternoon, everyone, or good morning, wherever you are. um, it's a great pleasure to be here. I want to thank first Andres and Miguel for the kind invitation to present here, and also to all the organizers of the European Society of Photobiology for a great conference so far. So our group have been interested over the years in DNA photochemistry, particularly understanding the electronics and structural factors that regulate the photostability of the DNA bases and their analogs, but also see if we can use this information, this fundamental information, to develop DNA derivatives that could act as a photosensitizer for therapeutic applications like PDT. So I'm gonna summarize here more than 10 years of work in two slides actually, because I want to move to the photosensitizer. But one thing that we have learned over the years is that the purine chromophore itself is not the photostable moiety of the DNA. It actually, when you substitute the DNA base, like in adenine, the position 6, I apologize, or the position 6 also with a oxygen atom in guanine, you have very fast intersystem crossing of the population from the excited state to the ground state. And so they are highly photostable, as we know in DNA. We also have seen that the sixth position is the key, because if you remove the amino group, for example, from guanosine, forming hypoxanthine, we still have a very highly photostable molecule and a biomolecule, as we all know. However, if you go and remove, for example, from guanosine, you remove the carbonyl, forming 2-aminopurine. Now you have a long-lived excited state species that both is a fluorescent molecule or can intersystem cross to populate triplet state and do reactivity. And so this suggests that the DNA photostability is not only related to the chromophore reserve, but is highly dependent on the functional group in the position 6. Of course, if hypothesis have a merit, you will expect that the pyrimidine molecules will observe similar phenomena. No? If you compare the purine chromophore with the pyrimidine chromophore, you can see that a purine base is no other thing than a pyrimidine analog with an imidazole group, right? In the 5-3 carbon position. So what you will expect is that substitution in the position 4, as in uracil, thymine, cytosine, and in this case, in the 4-pyrimidine, will create highly photostable molecules that can be used for in DNA. And while if you don't substitute in the position 4, you create highly photoreactive molecules. So, of course, you would not like to have the pyrimidine or these pyrimidones incorporated in DNA, because they can act as a photosensitizer. And so, so that's the fundamental knowledge. So of course, in DNA, there's not only the photostability, but then forming hydrogen bonding. So the functional group in the position 2 play a role in forming waxon and cream base pairing. Okay? But in terms of photostability, which in the prebiotic era, when there was no ozone layer, using these molecules as the building block, we think that substitution in position 4 was the key to maintain them and move on to polymers. And so, as I mentioned in the introduction, can we use this information to generate modified DNA bases and RNA bases for photodynamic therapeutic applications as a photosensitizer? And the first experiments that we did almost more than 10 years now ago the first thing that we did is that we said, well, if the position 6 and 4, 6 in the purine bases and position 4 the thymine bases is key to control photostability, let's make a single atom substitution and see what happens. Okay, so we replace the oxygen by a sulfur atom in guanosine and in thymidine, as you can see here, and we see dramatic changes. So as I mentioned before, 6-thio-guanosine have 0 triplet yield, highly photostable molecule, right? When you change a single atom, you now produce a extremely good sensitizer, a triplet yield of 80%, a single oxygen yield of almost 15%. In the case of thymine, you can increase the triplet yield to 85% and the single oxygen yield to 42%. So that's a dramatic increase in photoreactivity. Not only that, but also we can displace the absorption spectra to the red by almost 100 nanometers, which allows now to selectively excite the photosensitizer if incorporated in DNA. So, of course, one of the concern is the absorption is in the UVA spectral region. So we thought, well, maybe if we change from sulfur atom to selenium atom, we can redshift the absorption spectra more and still have a high photosensitizer. We show that that's actually the case. We can actually displace the absorption spectra, but not enough. It still stay in the near visible region of the absorption spectra. However, we can increase this is the so-called penetration depth in a tissue. We can increase the penetration depth by almost 90% by a single atom substitution. And I will mention in a minute why we are focusing on small substitution. But let me just throw it there, is because what we have found is that when we make this minor single atom substitution, the enzymes in the cell cannot differentiate between a normal DNA base or RNA base and the photosensitizer, and so they get metabolized and incorporated in DNA or RNA. So we have additional selectivity, not only by being able to selectively excite, but we can also incorporate these DNA bases analogs in the skin cancer cells and then selectively excite them. And so that's why we have been spending so much time on these molecules. We also say, well, what happens if we change the position of sulfur substitution? And so an experiment that we did is that we substitute in the position 4, as I showed you before, position 2 of the thymine base, or we double substituted the thymine base. And you see shown here in the absorption spectra, the black spectrum is thymine, it's just the natural DNA base. When you substitute in the position 2, there is an effect, but it's not a dramatic effect as when you substitute in the position 4. As we mentioned before, the position 4 seems to be more important for controlling the photoreactivity. And then when you substitute both, then we can redshift the absorption even further. So now we can actually have this double substituted thymidine base and excite at 400 nanometer as a photosensitizer. So we have done all the spectroscopic experiment. We see high population of the triplet state. In this case, is close to 100%. We are conservative because of the methods that we use, a comparative method. We report 90%, is plus minus 20, so it's very close to 100%. We have a 50% single oxygen generation, something that is not as relevant for this talk, but is still was very surprising for us and important from the fundamental point of view, is that all these molecules intersystem cross to the triplet state in hundreds of femtoseconds. So these are highly efficient intersystem crossing molecules. We understand the quantum mechanics of that. I will not be talking about that here today, unless you ask. But that was a very surprising, because these are fully organic molecules that show ultra-fast intersystem crossing to the triplet state. And so we have been focusing mostly on this double substituted because it's the one that is redshifted the most. And as I say, show remarkable properties for photodynamic therapy. But also we have studied the fourth IO timing in significant details. And so just to show now the some experimental results demonstrating that in fact these molecules can be used as an active photosensitizer. We perform confocal experiments using skin cancer cells line. These are basal cell carcinomas, skin cancer cells. We have been focusing here particularly in squamous and basal skin cancer cells because there are in the upper layers, in the dermis and the epidermis of the skin, where the sensitizer can penetrate and can do damage. They can only be excited at 400, so we cannot go deep in tissue. But as shown here, so what you see here is using a fluorescent dye. So this is the nucleus of the cell. And in the panel A, what you see is just the nucleus looks spherical, round shape. And that's what you will expect for a healthy cell that is alive. And then when in panel B, when we shine the cells with the light that we use, which is a 395 nanometer, the cells do not die, so they stay healthy and alive. So which you don't is where you want. If you add the photodrugs only and do not put light, the cell stay alive for a relatively low molar concentrations. Of course, if we increase the molar concentration to a point, then the photodrug itself can be toxic. But if we control the concentration, we work on the conditions that there are not high toxicity, then what we see is that if only when we incorporate the photodrugs plus the light, we efficiently kill the cells. We can quantify, apologize for that, we can quantify the efficiency of cell death. And so with a small dose, this is equivalent like a 3 minutes of a low intensity UV light exposure to the tissue. We can significantly reduce the proliferation rate of the skin cancer cells. We also have tried to understand in more detail the mechanisms, whether this is single oxygen generation or reactive oxygen species that are participating, or a combination of phenomena. So we have performed a confocal microscopy experiment using now this dye as a fluorescent reporter of reactive oxygen species. As you can see here, when you have both the light and the photodrugs, and using two different photodrugs here, 2,4-dithiouresil and 2,4-dithiothymidine, both of them generate a significant reactive oxygen species, and so at least part of the photosensitization event occur through reactive oxygen species. And I want to mention here that we are using also for double substituted uracil, because then we can target RNA. And in particular, we are interested in targeting mRNA. So see if they can be metabolized, incorporate, and then we have a Trojan horse, as Miguel would say, an internal photosensitizer that can do damage to the cells. I want to also mention and also to plug my next seminar tomorrow, which we have incorporated these thiobases in DNA synthetically, and we have seen that when we incorporate them in single-stranded DNA, we have a significant reduction, tenfold reduction in the single oxygen quantum yield, which suggests that there is the triplet state itself can react intramolecular and form damage, which is important and important for us because many tumor cells are deprived of oxygen. So singlet oxygen generation is not efficient. or you can consume the single oxygen and eventually the photosensitizer will not be able to perform. So we will show, if you come to my next presentation on tomorrow, that this photosensitizer can actually do ultra-fast 2+2 cycloaddition reaction, forming analog of the 6,4 photodimer, which is highly reactive and can damage DNA. And so these photosensitizers have a dual mode of action. They can, if they are in the cytoplasm, they can use single oxygen generation, reactive species. If they get incorporated and metabolized, they can still also be very effective and do damage. We also asked the question of whether the double substitution of the purine base can be used as a potential photosensitizer. And so we developed the double substituted purine chromophore shown here. It also absorbed in the 350, 390 region. And we were very surprised that the photosensitization efficacy is almost double of that of the double substituted thiothymidine and thiouracil. And we were also, we did similar cell experiments, including reporters of single oxygen. We found out that the correlation of the high efficiency of the photosensitizer is not related to single oxygen generation. So this is not a better single oxygen generation than the other two thiobases. And the only correlation that we have found is that it's related to the lifetime of the triplet state. Okay, so the longer the lifetime of as this case is almost twice as longer lifetime as in the case of the double substituted pyrimidine bases, the more effective photosensitizer is to the skin cancer cell. And so we are continuing exploring that experiments. Still, the elephant in the room is, well, these are photosensitizers in the UVA, near visible radiation. So we have been actively trying to find ways to shift the absorption spectra to the red, but without making major substitution to the chromophore so that the cell can still metabolize, incorporate them into DNA and RNA. So the next sequence of molecules that we study are these two shown here. They now absorb all the way to 410 nanometers, so we can use 400 nanometers for excitation. We show the same that when you have the thionation in the chromophore, you form highly triplet yields, high single oxygen yield, and we can also effectively damage the skin cancer cells, also generate single oxygen generation. And so those we are studying those molecules actively in our group, different skin cancer cells, but we wanted to do molecules that can be excited by visible and near-IR. And so we recently, in collaboration with Han Xiao from Rice University, we decided to see if we can generalize the photochemistry of this thionation process. And so we generate this family of thionated compounds that absorb all the way from the visible to the infrared. And today I'm going to focus on these two, which absorb in the infrared because of time. But we have studied the photochemistry, fundamental photochemistry of this compound completely. We understand also the theoretical mechanism, how the single oxygen, the triplet state is generated. These two molecules, as you can see, they both absorb in the near IR, and they populate a highly reactive triplet state. So most of the population go in both molecules to the triplet state. The single oxygen is high, and so they have the potential to be good photosensitizers. Again, heavy atom-free photosensitizers. And so recently, Xiao performed photosensitization experiments similar to the ones that we have performed before, but now with HeLa cells using both type of lights, either just continuous white light for 400 to 700 low intensity light, or using a laser light at 730 nanometers. And so you can see that they are highly efficient in, in, uh, in, in reducing the proliferation rate of the cells. As you can see in this confocal image here, when you have both the drug and the light, they light up uh, the, the reporter, and you can see that cell death is significant. Singlet oxygen generations also play a role. So these sensitizer work more effectively for tissue that is oxygenated and there are oxygen pressure. But these are highly efficient photosensitizers. Yeah, so I just realized that I'm not using my most correct presentation, but I have one or two slides that I wanted to show, but I will stop there. The conclusions are that we can use organic thionation as a functionalization of biocompatible organic molecules to generate highly efficient photosensitizers. And we are actively currently working with the medical school and the dermatology department to try to push this chemistry further. With that, I want to thank everyone for your time, and also my group members that did the work. Those that are shown here in the top have worked in this particular research project. And I also want to thank NSF that have been kindly supporting our research in the last 15 years. And now with that, I will be more than happy to answer any question that you may have.